What?
I said, Mary always says to remind you that we're doing it for her and not me.
It has nothing to do with him. Say, we like her. We may never give her back. Huh, Ruby? Did you tell him to wish Jake happy birthday?
Oh, I forgot. Okay, it's your turn. Yes, I promised you a dog treat tonight. He went upstairs, so she was going to chase upstairs after. So I pulled her off the stairs. Oh, it's not your favorite moment, was it? Okay. Let's get you.
Let's see if we get more students, because right now we don't have very many.
I'm here.
I know you are, but like if you look, there's like 7 participants, and so 6 of us. I think we just lost someone. I'm guessing it's an internet issue.
It's really windy everywhere, so I don't know.
That's maybe it, because yeah, it's cold out there. Wasn't that cold yesterday?
I nearly blew away when I went to Walmart.
Oh, I believe it. There's some more. All right, let's jump into this. So this is Wiley Lab Exercise 29. So this week, you're going to do the questions for the lab exercise 29 and the virtual lab of blood pressure. And you'll see this PowerPoint isn't very long. I'm trying to remember, I think it's 37 slides approximately, 41. Okay. So arteries, first, by definition, what's an artery? By definition, what is an artery? Talked about it earlier today. What is an artery?
A vessel that carries blood away from the heart.
Right. Vessels that carry blood away from the heart, and veins are going to bring it back. So major sequence here. Arteries are going to become arterioles. Arterioles are going to regulate the blood flow into capillary beds. Venules are going to drain capillaries. Venules will merge to become veins. So arteries carry blood away from the heart. Arterioles are going to regulate how much blood goes into capillary beds. Capillaries are where the real action happens. This is where exchange is going to occur. Bear with me, guys. Give me 1 second. I apologize. So arterioles regulate the blood flow into capillary beds. Capillaries are where the real exchange, we talk about how blood brings in nutrients and it eliminates, it removes waste. Where does that exchange occur? It doesn't occur in the arteries and it doesn't occur in the arterioles. It's going to happen in the capillaries. So the capillaries are what are going to bring nutrients and oxygen to the tissues and remove carbon dioxide and waste. This is where the exchange between the tissues and the blood is going to occur. Venules are going to drain the blood from the capillaries into the veins, and the veins are going to carry the blood back to the heart. Remember from earlier today, pulmonary versus systemic circulation. Little different diagram.
Can you repeat?
Go ahead.
I'm sorry, can you repeat that about the venules?
Venules are going to drain the blood from the capillaries into the veins, and the veins are going to carry the blood to the heart.
Okay.
This is a little different diagram, but it's the same idea as what we saw this morning, in that we have pulmonary circulation versus systemic circulation. Pulmonary circulation is taking it from the heart to the lungs, from the lungs back to the heart. And this is the place where the arteries and the veins are going to switch places with what carries oxygenated blood and what carries deoxygenated blood. So in this case, the pulmonary arteries are carrying oxygen poor blood, and the veins are carrying oxygen rich blood. This idea right here, this connection between the different vessels, is the same in both the pulmonary and the systemic circulation. When we look at the major amount of blood, and you have approximately 5 to 5.5 liters of blood per person. As it's allocated, most of it is in the veins. 84% of it is in the systemic circulation, 9% in the pulmonary circulation, with 7% of it going to the heart. Remember the coronary circulation that we talked about this morning. The rest of it is systemic. So pulmonary 9, heart 7, systemic is 84. 15% in the arteries, 5% in the capillaries, and 64% in the veins. I'm going to bring something up right now. We'll see it again later. But if you look at those percentages, where is the most amount of blood found? Where's the biggest quantity of blood found? Somebody tell me.
What was the question?
Where's the biggest quantity or percentage of blood found in the body between the different vessels?
It said systemic system, but I feel like that's not the right answer.
You're right, but where in the systemic, you're right, but where in the systemic circulation is it found? That's what I'm asking. Where's the biggest percentage found in the systemic circulation? My question may not be very clear here. It's found in
the organs?
Well, no, within the vessels. Within the vessels.
Sorry.
Like the capillaries, you mean?
Nope. Go look at the numbers on this other slide. And it may be that my question is just not a very good question.
The vein.
The veins, right. This is why our veins are called the capacitance vessels. Because when they work, they're kind of where blood can pool when it's not necessary somewhere else. They're flimsy. They're flimsy in their structure. They're flimsy in their musculature. They stretch really easily. Or actually, they don't really stretch really easily. They just they're flimsy. That also allows them to be kind of low blood pressure and carry a large volume. I think, is it capacitance? I think that's right. Yeah, I think that's how it's spelled. So, and I know this is a little blurry, but it's not too bad. Within each of the different types of vessels, except for the capillaries, there are 3 different tunics. What's tunic? What's a tunic?
Are we talking fashion? No, I'm just kidding.
Well, yeah. What's a tunic?
I feel like I sort of have a tunic on.
Right.
Covering.
Well, don't make it too difficult, Becky. Yes, it's a sweater, it's a covering, it's a coat, right? So the 3 different tunics in the vessels are coverings. The most internal one is the tunica intima. If you think about your intimate clothing, it's the stuff that's the most internal right up against your skin. tunica media in the middle, tunica externa on the exterior. We also, another thing that we call these, bear with me here. Tunica intima, depending on where you look. Intima, yeah, is also the interna. Tunica interna, meaning the most internal layer. Tunica media is the same for both. It's always the same. I've never heard it said differently. Tunica externa can also be called the adventitia. The way that I remember the tunica adventitia is if you live more on the periphery of a town or more on the outside of a town, you're more likely to take adventures. So tunica adventitia. So tunica intima, most inner layer, tunica media, the inner middle layer, and tunica externa, meaning the outer layer, or tunica adventitia. These are the same for all of the vessels, except for the capillaries, which just have a tunica intima. So looking at the very beginning, here we have our elastic arteries like our aorta, like our brachiocephalic trunk, like our common carotid artery and our left subclavian artery. These are highly elastic arteries. Oh, and you know what? I was going to grab something before I sat down. Just a sec. Bear with me just for a moment. So elastic arteries or conducting arteries right next to the heart. And as we move further away from the heart, we have muscular arteries, meaning more muscle and less elasticity. Then the arterioles, which in comparison to the other vessels have more muscle relative to the other tunics. This is going to be important in a moment. Capillaries have only the tunica intima layer, which is only the endothelium. Do you remember what did I tell you this morning about the endocardium and the inner lining of the vessels? Go ahead.
I was going to say, what was that again?
What did I say this morning about the endothelium that lines the inner part of the heart and the inner lining of the vessels. So the most inner lining of the vesicles, the tunica intima, is continuous with the internal part of the heart and throughout the vessels. The same inner lining that you see in the heart is the inner lining of the vessels. It's one continuous layer throughout all the vessels up until the vessels return back to the heart via the vena cava. So arterioles, relative, more muscle relative to the other layers. When we move into the veins, we have very little muscle and more tunica externa. More tunica externa. If you look, here's the tunica externa on the arteries and on the arterioles. Look at its relative thickness for the veins. It becomes a much thicker tunic in the veins in comparison to the other tunics. The elastic arteries are the, why would they need to be elastic? What would be the point of having elastic arteries right next to the heart? Remember what it means when the heart contracts. So the heart contracts, and what was that called when the heart contracts? What was a heart contraction called? What's the scientific term?
Systole?
Systole or systole. So your heart about two fists. Bear with me here. About 2 fists. Okay. It fills up it, and then it goes poof, and all of that blood rushes out through the arteries. Goes poof. What's going to happen in the aorta at that point? You notice that the aorta is not that big compared to the heart. What's going to happen at that point? Goes
Like going to shoot out.
It's going to shoot out. And how big is your aorta compared to your heart? Look at that picture. How big is it compared to your heart?
It's like a third of the size.
Yeah, it's maybe a third of the size, maybe a little less. It's probably more like a fourth. So all of a sudden there's this major volume that comes into the aorta. It's like, and one thing that you need there is you need some elastic, because that aorta is going to expand. There's no, water only flows so fast, right? Water, AKA blood, only flows so fast. And so it's, for a second, the aorta is going to go. And because it's an elastic artery, it's going to go, it's going to recoil back just like your elastic bands do. It's going to stretch out, and then it's going to elastic recoil and come back. So pretend I have a balloon. We're going to use this a lot over the next 2 days of class. So I have this balloon like this. Becky's not even looking.
I'm sorry.
Here I'm trying to do a good demo and she's not even watching.
Well, my husband needs to come home and take my son to the CAT scan. So I was in the middle of that.
That's no excuse. Okay. Grateful laughs. All right. So awesome demo. We have this. And so all of a sudden you get a great, the heart contracts and it shoots the blood out. Okay. All of a sudden my aorta went from being that to that. Well, it's going to recoil and shoot it out towards the other vessels. That's why they're elastic arteries. You got to have elastic arteries because that recoil is going to push it forward. If we had it shoots out and it's going to go out to the other vessels, but that recoil actually helps push it forward too.
Excuse me, I just thought of something.
Go ahead.
You were talking about like doing an experiment and stuff. When are you doing the, what is it, like the urine analysis?
And probably a couple weeks.
Okay, I just forgot about it and I only remembered now, so I just wanted to bring it up. Sorry for interrupting.
Yeah. No, it's all right. I will I need to get I know I have a lot of people's addresses. If you haven't sent it to me as an email or text. I need to write them all down. And I will probably send it out over spring break for you guys. But let me, somewhere in there.
Our physical address?
Yes, your mailing address.
Okay.
And the reason is so I can send you something.
Okay, no problem. I just wanted to verify. It's okay.
Yeah. I'll shred them as soon as I mail them out. I'm gonna shred all your addresses. I don't keep them around.
Yeah, I have an open case right now with the Fed, so.
Well, you know what, Casey? Maybe you should.
I'm joking.
You should quit getting in trouble.
I'm joking.
Darn it all. Some people's children. All right. So lots of elasticity in the elastic arteries. More muscle in the musculature arteries relative to the others. These are also the distributing arteries. Lots more of these. And these elastic arteries right next to the heart. Muscular arteries a little further away, the distributing arteries. The arterioles, the capillaries, the venules, the medium-sized veins, and the large veins. Why do the tunic components and widths of those components vary between the different vessels? If you look at them, the tunica media in the veins is much smaller compared to that in the vessels. Why is that the case? Why would that be important? Why would we have elasticity over here, but not so much over here?
Is it because more blood is pumping out of the one side?
Yep. I mean, that's it. And why, if you look at the lumens over here, here's the lumen of the arteries. Look at it in the veins. Why is the lumen so much bigger in the veins? Hold on to the question just for a minute. We'll come back to it. So remember the relationship between arterioles, capillaries and venules. Arterioles are going to regulate the blood flow into capillaries. They have more muscle relative to the other layers because they're going to be constricting and dilating. Bear with me. Constricting and dilating based on how much blood flow needs to go into that capillary bed. This is where the real deal comes. This is where it becomes important that we only have like 5.5 to 5.25 liters of blood in our whole entire body. And if you think about your net volume of your body, you're probably looking at easily, I don't know, I'm a pretty small person. So I'd say maybe my whole mass volume, if I measured me all together, I'm probably 8 gallons, maybe. So I only have 5.5 liters, and you have vessels everywhere. So that means that some of your vessels have to have very low flow while others have high flow if you're going to be able to distribute the blood correctly, right? If you have 5.5 liters max, and I was like, Kevin, I can sometimes see his face. He looks like he's a pretty big guy. He's not really little, but he's fairly big. He's not really big, but he's somewhere in the middle. I don't know how tall you are, Kevin, but you don't look like you're really little. So we'll say that he's maybe, I don't know, all volume wise, he's about 10 gallons. And he's got 5.5 liters. So some capillaries beds are not going to have as much blood running through them as others. In order to distribute it so that the most amount of blood is going to the areas of your body that have the most amount of activity at any given time. This is regulated by the arterioles and what we're going to talk about, the meta-arterioles, is how much blood goes to what area at any given moment. So the tunica interna, endothelium, and basement membrane. Remember, this inner layer here is the same as the endocardium. The same exact thing, and it's continuous throughout all the vessels. The tunica media mainly is smooth muscle. Tunica externa has more connective tissue, allowing that vessel to stay connected. You notice that when you have your veins and your arteries, they don't slip around a whole lot. You know, you can, I'm a pretty veiny person. Bear with me here.
Why do some people have like spider veins? Do you know what I mean? Like they're like very prominent veins on your thigh. My mom and my grandma.
We will get to that. We will get to that, like Lauren, it's coming.
Yeah, I have them really bad, and all of the women in my family have them too.
I think sometimes it's a genetic issue. I don't think, you know, I heard someone told me it came with pregnancy. Well, for someone like me who's never had a pregnancy.
Yeah, I don't want that to be true.
Well, I mean, yeah, for someone like me who's never had a pregnancy, why do I have them? I think there's a lot that, but I'll get to that one. Bear with me. So this vein right here, this vein right here, you notice it doesn't, I mean, regardless of what I do, it doesn't slip and slide, right? It doesn't like slip over here. It doesn't slip over here. It's not really tight either. I can compress on it and allow it to fill up with blood, but it doesn't really move. The tunica externa is anchoring it in place. That's why your vessels don't tend to move very much. Is because they're being anchored in place by the tunica externa. But either we'll get to it in this lab, Lauren, or we'll get to it in lecture when we go through blood vessels. But we will talk about that. Yeah, bear with me. We'll get to it. We may get to it today. So capillary beds, arterioles, meta-arterioles, and precapillary sphincters. Here's my arterioles. When they contract, when they're dilated, blood is moving through them. Sphincters are opened, and the blood can flow into the capillary beds via this meta-arteriole, which is a thoroughfare channel running through. We have precapillary sphincters that determine how much blood is going to go out into the capillaries. So blood is going to be regulated at the arterioles, and it's going to be regulated at the meta-arterioles via the precapillary sphincters. So how much blood goes out into the capillary beds is determined when it's moving from the meta-arteriole into the capillaries. How much blood moves into the capillaries depends on the constriction or dilation or relaxation of the precapillary sphincters. If the precapillary sphincters are closed, the blood flows right through the meta-arteriole and right through into the venule. If they're open, it flows into the capillary beds, depending on how much blood is needed to that area at that given moment. Meta-arterioles. So blood flows into the arteriole, flows into the meta-arteriole, through the precapillary sphincters into the capillaries. Bear with me here. So arterioles, to meta-arterioles, actually, yeah, meta-arterioles. I think actually just, I think it's no, it's arterioles. No, I think it's 2 A's. All right, bear with me. I have to look. I can't remember. Just doesn't sound right. It's 1. So I'm sorry, I have to, you gotta love this whiteboard. It doesn't let me just add to something I already started. Okay. So arterials. Meta-arterials. Meta-arterials. To precapillary sphincters. To capillary beds. To capillaries. Remember, the meta-arterials are thoroughfare channels. Thoroughfare channels. When the precapillary sphincters are closed, precapillary sphincters are closed, no blood flows into the capillaries. Or it's very minimal. When they're open, blood flows into the capillaries. Questions? So capillaries, pretty amazing things. If you ever get a chance to see a Body World exhibit, it's a international museum type traveling museum. And I went and saw it when I was living in Idaho. I was teaching anatomy and physiology. That's where I first started. And I went down with a bunch of the teaching assistants for anatomy and physiology, and we went down to Salt Lake. It's about a 3-hour trip. We went down there and visited the Body World exhibit. And one thing that they had was they had a body where they had filled up all of the capillaries, all of the vessels, and then dissected away everything else. Dissected away all the extra tissue, leaving only the vessels and including the capillaries. And the result is that the structure takes on the same shape and structure as the body itself, because you have capillaries everywhere. There's very little structure to your body that doesn't contain capillaries. And so if you dissect away everything except for the capillaries, it takes on the same shape and size as you are. A lot of cancer drugs are looking at stopping blood vessel growth, capillary growth specifically, because if you stop capillary growth, you stop tumor growth. Because they're so necessary. It's where growing metabolizing tissues get oxygen and nutrients and eliminate waste. And without them, that tissue won't survive or grow. Questions? So capillaries, remember, this is where I told you, this is where the real business of the blood happens. Extremely small diameter, composed of a simple squamous endothelium and a basement membrane. Think of this the same thing as they are composed solely of a tunica intima. That's all they are. Because if you remember the tunica intima, which was the same thing as the endocardium, thinking to what we were talking about this morning, it's the same tissue components in the same structure. So here's our capillaries, and they're so small that the red blood cells, the erythrocytes, are going to be moving through them almost always single file. Always moving in a single file, because that's the only way they can move through. High resistance, high vessel resistance, and single file. In a lot of our vessels, so it's single layered. So remember what simple squamous epithelium means. So that's flat, one cell layer thick with a basement membrane. You have intercellular clefts, so clefts in between the cells, and fluid exchange. Question? We have three basic types of capillaries. Continuous capillaries, which is where the basement membrane is continuous. And there's some tight junctions in between the endothelial cells. That's our continuous vessels. These are the most common vessels. Most common capillaries are these continuous, where the basement membrane is continuous. We have fenestrated, fenestrated. Fenestra means holes. Fenestrae.
Fenes means what?
Holes, like H-O-L-E-S, holes.
Oh, hole. Okay. Thank you.
And so these are where there's small holes within the endothelial cells and within the basement membrane. So the main, let me finish this. Sinusoidal, what does sinus mean? A big space, big gap. Think about when you have a sinus infection, the big cavities within your facial bones all of a sudden feel like they're swelling with fluid. Sinusoidal or discontinuous, meaning that there's big holes in the endothelial cells, not just little fenestra, little tiny holes. These are big holes, and big holes in gaps in between the cells, and has a discontinuous, so a very choppy, rather than just having a holy basement membrane. Now the basement membrane may or may not even be there. If it's there, it's very flimsy and very weak. So we have 3 different types of capillaries, and they're located in different places depending on what's needed at that time, what's needed in that area, how much perfusion of the blood is necessary there. See, you find sinusoidal capillaries in areas where there's a lot of filtering, your kidneys, your spleen, your liver. Fenestrated tend to be in, where would you find fenestrated? I can't remember. But continuous are the most common. What is transcytosis? What does cytosis mean? What does cyto mean? What does cyto mean?
Cell.
Cell. What does trans probably mean? You've heard of, like, you transfer. What does trans probably
The
mean?
moving of a cell?
Or moving of something through a cell. So transcytosis is moving something across the cell, from one side of the cell to the other side. So some terms to remember here: exocytosis, endocytosis, and transcytosis. What's exocytosis?
Leaving the cell.
Something leaving the cell, right? So something leaves a cell. What's endocytosis?
Within the cell.
Or actually, you're close. What else could it be? So bringing
Going in the cell.
Say it again.
Going in the cell.
Yeah, coming into the cell. So endocytosis, something comes into the cell. Then it has to move across the cell, and then it moves out of the cell. That's transcytosis.
Excuse me.
Bear with me here. So here's my cell. Here's my cell. Something moves into the cell, endocytosis, moves across the cell in vesicles, and then it leaves the cell via exocytosis. Questions? So in the blood vessels, things are going to move single file. Excuse me, in the capillaries, things are going to move single file. Here's a longitudinal section through a capillary. Here's a cross section through the capillary. You guys give me 1 second. I apologize. Ruby, you better not be back there. Come back. I'm sorry. Give me 1 moment. My mom's asleep and we left the door open. You get back here. You don't need to be back. Ruby, come on. Out of there. I'll take care of her. Okay. Apologize. Okay. So this is looking at a cross section through a capillary. This is a longitudinal section. What tissue is this? What kind of tissue do you think that is?
Is that cardiac?
It's cardiac or skeletal, right? Because it's striated. So here's some more. Single file. What kind of tissue is this?
Which one?
This one right up here where my in the top right corner.
That's definitely cardiac because of the.
It's not. Look at it. Look, is it muscle? You sure?
No, it's not muscle because it's not striated.
Well, and there's other reasons it's not muscle. Well, what other type of tissue could it be? What other types? You know, it's not epithelial tissue. It's not
I think
bone.
it's at the top. It's a capillary.
Well, it's a capillary, but what kind of tissue is this? That's the capillary is moving through. This is actually looks more like an arterial.
A smooth, like what is the tissue that's smooth? And then
cartilage?
No.
Connective?
No.
What do you think it is, Kevin?
Yeah, I was going to say
Epithelial. Process of elimination.
It's not epithelial because there's too much extracellular matrix in there for it to be epithelial. You guys, I think it's areolar connective tissue.
Yes.
Because of all the space in here. You guys, some of these pictures, I'm trying to do better by getting some pictures right from your manual. And I realized that I could copy and paste them right from your images. This is one of them. So if you look here, here's your arterial. Here's your individual capillaries. And if you look, the little black specks in here, I'm guessing those are red blood cells. And if you look, they're moving single file through. Here's some more pictures. Intercellular clefts. Remember when we're looking at intercellular clefts, intercellular junctions right here between the endothelial cells. Here's looking at a cross section through a capillary, intercellular clefts. And vesicles. Here's the lumen of the capillary. vesicles involved in transcytosis. Here's one pinching off. Here's one moving across. Tight junctions. This is an intercellular cleft, and with that, there's tight junctions. Wiley may not be my favorite choice when it comes to publishers, but they have some neat pictures. Questions about capillaries? So since capillaries are where the real business occurs, we have opposing forces that are going to regulate filtration and absorption in capillaries. And you know what? I'm going to do something. I told you when I'm teaching this course, sometimes I go by the seat of my pants and I'm just kind of rolling with it. But I'm going to find something, because this is something that I want you to understand and understand it well. And we have time. Okay. So there's opposing forces that are going to be involved in regulating movement in and out of the capillaries. And I want you to get a really good grasp of this. So give me 1 second here. Even though this is 20 minutes long, let's watch this.
In this video, we're going to have a look at capillary exchange.
So capillary exchanging, meaning as things are moving in and out of the vessel and moving in and out of the capillary.
So capillary exchange happens at capillary beds.
You listen to how he says capillary and how I say capillary, or how he says capillary and I say capillary. It's the reason we don't make fun of people's pronunciation. Because I'm not going to go against this guy with regards to what he knows and what he doesn't know. Let's see. Maybe. It sounded like a good idea to watch it. Give it a second. Let's see. Maybe not.
In this video, we're going to have a look at capillary exchange. So capillary exchange happens at capillary beds, and this is the exchange of bulk fluids out of the capillaries. And once this fluid has exchanged out of the capillary, it's able to, by the process of diffusion, hand over the oxygen, hand over some of the nutrients that's required for the tissues, and also the tissues need to hand over to that fluid carbon dioxide and also some metabolic waste products like urea, for example. So basically, we have a...
What's diffusion?
Something that goes down a concentration gradient from high to low.
Right. Okay. So that's the way this is all being regulated. Depending on where the pressure, where the concentration gradient and the pressure gradient is highest.
You've taught me things.
I'm glad I'm teaching you something, Claire. I'm glad.
You need to remember this. When the heart contracts, when that left ventricle contracts, it's pushing out oxygenated blood, which has nutrients in it, out of the left ventricle into the aorta. And remember where this blood goes. Once it goes out by the aorta branches into all of its aortic branches. And these continue to branch like a tree. Now, as these arteries, because it's leaving the heart, as they branch, they get smaller in diameter, but they become more numerous, okay? And all of these branches end up feeding all the tissues of the body. Now, every minute, that heart's going to contract and relax, contract and relax, contract and relax. Every minute, this is going to happen around about 72 times, okay? So you have around about 72 beats of your heart per minute. Every contraction pushes out approximately about 70 mils of blood. So 70 mils pushing out is called your stroke volume. Every contraction pushes out 70 mils times 72 beats in a minute means that in a time span of 1 minute, your heart will push through 5 liters of blood. Okay, so this is on both sides of your heart.
So when he's saying that, what do we define that as being? How much blood is? What do we call that when we're the term that says how much blood is being pumped out of the heart per minute from last year?
Heart rate?
Nope.
Or stroke volume? Stroke volume?
You're close. Times them together. What are they?
Something output? No.
Yep. Yep.
It is something output.
Cardiac output.
Right. So that's what he's saying right there.
So remember, the left-hand side of your heart deals with oxygenated blood and delivers it to the whole body. Every minute, the left-hand side of your heart will deliver 5 liters of blood.
Systemic circulation. Systemic circulation. Pumping from the left side of the body.
The right-hand side of your heart deals with deoxygenated blood. And therefore, this blood...
Pulmonary circulation. So left side of the heart, systemic circulation. Right side of the heart, pulmonary circulation.
Needs to get to the lungs. And again, every minute, this side of your heart will push out 5 liters of blood. So if we just have a look at what I've drawn here on this side of the whiteboard, you'll see we've got the heart. If we have a look at the left-hand side of the heart, when it contracts, pushes blood out of the aorta branches into all of its aortic branches and then further branches into smaller arterioles. And these arterioles will then lead to capillary beds. And these capillary beds could be capillary beds of the coronary arteries, cerebrovascular capillary beds of the gastrointestinal tract, of the renal system, of the musculoskeletal system.
You guys, even, you know how I said I wanted to do lecture today? Even if we take all this time to talk about capillary exchange, get this down. Because we're going to talk about it in lecture. And if I can make up for the time then, that wouldn't be a bad thing. Because this is important when we're talking about blood vessel physiology. So focus on this. If we don't get to any more of the heart today, that's okay. This is important stuff. It's important for a lot of reasons later.
And also the skin. Okay, these are just potential places that this blood could go through. Now remember, as it gets to the capillary bed is the site of exchange, and that's exchanging gases, oxygen and carbon dioxide, and also nutrients and waste products. Then on the other side of these capillary beds, we have veins. Okay, veins will always go back to the heart, and you can see they all come together, These veins will get back to the right-hand side of the heart by the vena cava, whether that's the superior or the inferior vena cava. Now to the right-hand side of the heart, it will contract and push that deoxygenated blood where? To the lungs to get oxygenated. And again, this blood's coming through to capillary beds of the lungs. If we look at this portion, so the blood that's going to the body, every minute, 5 liters. If we look at this.
So of all the vessels in the body, where's the major site of exchange for blood? Of all your types of vessels, of blood vessels, where's the major site of exchange?
Capillaries.
Right.
Portion going to the lungs every minute, 5.
Is it different? Is it the same? Take a guess. Is it the same capillary versus systemic circulation? That it's always going to be the capillaries as being the site of exchange. Yes, that's always the site of exchange. When we talk about the lungs, we're going to talk about how the lungs look like they're coated in blood. And you'll see it because I told you I have some fresh lungs. They look like sheets of blood because there's so many capillaries surrounding them. Because what's the point of the lungs? The oxygen exchange.
But here, this blood is partitioned to various tissues. Now, why am I bringing this up when we're talking about capillary exchange? Because when the blood's pushed out the left-hand side of the heart, it's under a particular amount of pressure, okay? And in one of the previous videos, I've spoken about blood pressure, and I spoke to you about the fact that a normal blood pressure is 120/8. 120 is the systolic value. So 120 is telling you that when the heart contracts under systole, this is the pressure that the...
So the systolic blood pressure is the pressure of the blood during heart contraction. The diastolic blood pressure is the pressure of the blood when the heart is relaxed. Systolic and diastolic.
The blood exerts on the walls of your arteries, 120 millimeters of mercury. The bottom value, 80, is the diastolic value, and this is the amount of pressure within those arteries when the heart is relaxing, okay? Now, saying 120/80 is important clinically, but to know what the mean arterial pressure is, I told you that you need to take the diastolic value and add 1/3 of the pulse pressure. Now, if you're unsure what that means, go back to the previous video looking at blood pressure.
I'm going to stop just for a second here. We'll see this again in a minute. But so what he's saying is the way you calculate blood pressure is mean arterial pressure. Shoot, and now I forgot what he said. 1/3. Okay, never mind. I'm not going to try this. That was bad. All right, just a sec. I knew what it was until just a second ago.
When the heart is relaxing, okay? Now, saying 120/80 is important clinically, but to know what the mean arterial pressure is.
So MAP, mean arterial pressure.
I told you that you need to take the diastolic value and add 1/3 of the pulse pressure. Now, if you're unsure what that means, go back to the previous video looking at blood pressure, and it'll explain it all. But mean arterial pressure is 93 millimeters of mercury. That's just saying that going through the arteries of the body, the mean pressure that the blood is exerting on the walls is 93 millimeters of mercury. And as this 93 millimeters of mercury starts to move through these various branches, the pressure drops. And by the time the blood reaches the capillary beds, It's no longer 93 millimeters of mercury. By the time the blood reaches the capillary beds, the pressure of the blood inside the capillary beds is 30 millimeters of mercury, between 30 to 35. Okay? Now, what does that mean?
So it's starting around 93 and it's going to 30. What do you know about pressure and flow through a hose? As the pressure goes down, the rate of flow goes down too. So blood is going to flow a lot slower through the capillaries, which we want that to be the case because that's where the exchange is occurring, and we need to give it time for that exchange to happen.
It means that blood's going to move through these small arterioles until it gets into a capillary bed. And you know capillaries are porous. That means they have holes in them. And you can see these little holes through this capillary bed, okay? Now, this blood's getting through, and by the time it reaches what we call, you can see one side of the capillary bed I've drawn red, the other side I've drawn in blue, because one side is the arterial end.
Video anymore? Are we not supposed to be seeing
the other side is the venous.
What were you saying?
Are we not supposed to see the video anymore?
Are you not seeing it?
No, I can't.
Good thing. All right. It's a good thing you're saying something to me. I think I know what I did wrong. Bear with me here. All right. Let me scroll back just a little bit.
The mean pressure the blood is exerting on the walls is 93 millimeters of mercury. And as
Do you
this
see it
93
now?
millimeters of mercury starts to move through these various branches, the pressure drops. And by the time the blood reaches the capillary beds, it's no longer 93 millimeters of mercury. By the time the blood reaches the capillary beds, the pressure of the blood inside the capillary beds is 30 millimeters of mercury, between 30 to 30.
I know I didn't. I didn't think I did that.
Now, what does that mean? It means that blood's going to move through these small arterioles until it gets into a capillary bed. And you know capillaries are porous. That means they have holes in them. And you can see these little holes through this capillary bed. Okay? Now, this blood's getting through, and by the time it reaches what we call, you can see one side of the capillary bed I've drawn red, the other side I've drawn in blue, because one side is the arterial end and the other side is the venous end.
So arterial end coming in this way, venual end or venous end this way. So the arterial regulating the blood flow into the capillary bed. Here's the venual end draining it, the blood.
Because remember, arterial blood is oxygenated. Venous blood is deoxygenated, which means that here we have that stuff coming out, and here it's no longer oxygenated. So blood's being pushed out of here, exchanging gases, so pushing out oxygen, taking in carbon dioxide, and here we now have deoxygenated blood, and this blood is ultimately leaving the tissue and going back to the heart and into lungs. So, as this blood's coming through, as it gets into the arterial end of the capillary, there is a blood pressure that's pushing out. And this blood pressure is, like I said, 30 millimeters of mercury. Now, remember, pressure that we measure in blood vessels is simply as the blood moves through the vessel, blood is pushing on the walls of those arteries. There's no holes.
What is that called when if it's pressure that's moving on the walls of the vessel or moving up against the walls of the vessel? What is that when you think of your garden hose in the backyard?
Filtration.
Oh, well, that's what's going to drive it. That's the filtration. But what is this pressure that he's describing right here?
I'm not sure.
What about the hydrostatic pressure? So this is just, you know, filtration is what's ultimately going to occur, Zena. But one of the major, but the pressure that the fluid is exerting against the walls of the vessel is the hydrostatic pressure. Does that make sense?
Yes.
Okay.
In the walls of arteries. They're not porous, so blood cannot leak out. Okay? As those arteries branch out even further and get smaller in diameter, they turn into arterioles. And again, arterioles don't have holes, they don't have pores, so blood cannot be pushed out. But it's when those arterioles branch off into these smaller capillaries, that's when they have these holes and these pores. And therefore, as the blood comes through and pushes on the walls, it can leak out. And so once we get to the capillaries, we now have 30 millimeters of mercury pressure pushing on the walls. And that means that this fluid, this blood fluid, is being pushed out of the capillaries. Okay? At 30 millimeters of mercury pressure. Now, the important point is this. You can see that there's little holes in the walls of the capillaries. These holes are of a particular size. Okay? Now, what is in blood? You need to remember what's inside of blood. It's not just a fluid. Inside blood, we have.
You guys, what are different things that are found in the blood?
Besides like erythrocytes?
Yep.
Like
what other things?
Iron.
Iron.
Calcium, sodium.
Calcium. What about
sodium?
Sodium? What about albumin? Fibrinogen. Lots of different proteins. What do you remember about osmosis?
Like concentration gradient?
Yep. So if you think about that, the more concentrated an area, if there's more fluid out here and less fluid in here, the fluid is going to move into the vessel, right? Just based on osmosis, fluid is going to move in. There's more proteins in here in the vessel and less proteins out here, it's going to move in. There's more fluid in here and less proteins out, and more fluid in here and less fluid out here, it's going to move out. Just based on the osmosis, looking at just keep thinking of blood as being a fluid. It doesn't matter that it's blood and not just straight water, but it's very much a fluid, and it has the same dynamics as the water running through your hose in your backyard.
Red blood cells. We have platelets. We have white blood cells. We also have what? Proteins. Okay? So all these substances of the blood is coming through. Now, these holes are too small to let white blood cells, red blood cells, platelets, and proteins out.
So all of the proteins, even though the fluid can move out here, all of the proteins are trapped in here. So what's going to happen at this other end? If the only thing that can move through the walls here is the water or the fluid, what's going to happen to the proteins? At this end, the proteins are going to be much more concentrated than they were here. And the result is that the fluid is going to want to move back in. Does that make sense? Just based on what you know about osmosis?
Yes.
Okay, they're too small. So you remember diffusion. Remember that in diffusion, if you have a concentration difference of a substance on one side of a membrane compared to the other side of a membrane, that this concentration difference will want to balance itself out. Okay, so if I can draw it up here very quickly, I would have a container, and I was to put a membrane in between.
I'll stop marking the video.
And I was to fill both sides up with water. And on one side of the membrane, I was to put some substances, some particles. And on the other side, I don't put any particles. Well, under diffusion, these particles on this side, so side A compared to side B, the particles on side A would want to diffuse from its high area of concentration towards its low area of concentration. Okay? But there's a membrane here, and this membrane will not let these particles through. So it cannot diffuse across from A to B, but there's osmosis. And osmosis is the movement of water through a semi-permeable membrane from an area of high water concentration to an area of low water concentration. What that means is basically this. These particles on side A exert a pull. Okay? They exert a pull of that water from side B to side A. So that means in order to balance out the concentration of both sides, these particles pull water towards them. Okay? What happens to the water volume on this side? Starts to rise up. Okay? So think about that in regards to the blood. We have the blood moving through, and it's exerting pressure on these walls at 30 millimeters of mercury. There's holes here, so the fluid can leak out at 30 millimeters of mercury. But we have substances inside that cannot move out. These substances want to diffuse out of the vessel, but they cannot. There is a semi-permeable membrane in between. So what that means is these substances exert a pull back in. They exert a pull because there's a high concentration of particles here and a low concentration of particles here. So they actually exert a pull. Now, of all these particles, I said,
when the particles are exerting a pull towards where they are, that's osmotic pressure or colloid osmotic pressure. When the fluid is pushing out, that's a hydrostatic pressure. So osmotic has to do with the osmolarity or the molecules within it. Hydrostatic is and that's having to do with moving things to wherever the particles are. It's a pressure being exerted by the particles to move things back into wherever the particles are. Hydrostatic is the opposite, moving fluid out.
Red blood cells, platelets, white blood cells, proteins. Which of these exert the strongest pull? The answer is the proteins. Now, the next thing is, which proteins? You should know what the most abundant protein is within the blood. It's called albumin. And this albumin protein is very important in eliciting a pull of the fluid back. Okay?
If you remember, albumin, the one he just said there, is the most dominant protein in the plasma, and it has the strongest influence on the osmotic pressure. With the ability to bring water back into the vessel.
Now, the strength of this pull, is 20 millimeters of mercury. Now, I want you to have a look at this. We have a push out of 30 millimeters of mercury and a pull in at 20 millimeters of mercury. When you have two pressures opposing each other, the stronger pressure will always win. But if you have two people pushing against each other and you say to one of them, push harder than the other person, that person's going to win. Okay? They're exerting more force. So if you have a look at these two, which one's exerting more force? The 30 millimeters of mercury pushing out. That overpowers the 20 millimeters of mercury by, what's the difference? 10 millimeters of mercury. Okay.
So the net, if 30 meaning pushing out, 20 pulling back in, therefore the net movement is going to be 10 millimeters coming out. So the net movement is going to be fluid moving out. Is that clear? Maybe. No?
Yes.
Okay.
Yes.
What that means is at the arterial end of the capillary, stuff gets pushed out. Okay? There's a pullback in, but overall, stuff gets pushed out. Now, of the stuff that gets pushed out, this is fluid. This is a bulk push of blood fluid coming out. And in
bulk push meaning bulk flow. That's one of the terms that I know is going to come up in your questions this week. Bulk push meaning bulk flow. Things are moving by bulk flow.
In this fluid, it has gases because they're small enough to get through in the fluid. And it has substances like glucose, they're small enough to get through these gaps as well. And then this fluid can exchange with the tissues. Now, as we move across from the arterial end, because remember, blood's moving through like this, it's coming through, pushing out. As we move from the arterial end to the venous end, I want you to think about what happens to this pressure. If you had a hose and you were to connect that hose to a tap, and you turn that tap.
What's going to happen to the pressure as it moves down? What's going to happen to the two different pressures?
Are they going to go opposite of each other?
They are. They are. So as it moves down, the osmotic pressure is going to become much higher, and the hydrostatic pressure is going to become much less.
Tap on at a particular pressure. So you turn it on, let's say you turn it on halfway. And you had a look at the hose. Let's say the hose is 10 meters in length. You look at the other end of the hose and water's squirting out that end at a particular pressure. Okay. Let's say you were to...
If you listen to him talking about hoses, you see why I tell you to keep thinking about it as a hose.
Now, put holes in that hose. So let's say every meter for the 10-meter hose, you put a hole, 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 and you have it turned on at halfway, and you have a look at what's happening. At those holes, you see water squirting out, right? Now let's say you have a look at the hole that's closest to the tap. The water's going to be squirting out of that hole at a particular pressure, at a particular height. As we move down, what do you think? So just think about what's going to happen to the pressure of that water pushing out those holes. Is the pressure going to increase or decrease? Is the water going to push out at a higher distance or less? Well, think about it. Hose is turned on. Water's pushing out at a particular height closest to, but because that water's leaving, the pressure drops distal to that. And the next one squirts out, but it's a little less. And then the next one drops. Little less. So the pressure decreases as you move down the hose because these holes are reducing the pressure as we're moving through. That's what happens in capillaries. So as we move from the arterial end, stuff's getting pushed out, so the pressure in here drops. And that means at the venous end, the pressure drops enough that it ends up being a push out of 15 millimeters. Now, these substances, such as, and I'm just going to draw the alcohol, is continually moving through. So it's still on this side of the venous end of the capillary. Okay, so these substances are moving through. So these proteins are still exerting a pull of the fluid back in. And this pull is still around about 20 millimeters of mercury. So have a look now. If you were to measure the pressures against each other, which one now wins? Well, 15 out, 20 in, the pull in now wins. The difference, 5 millimeters of mercury. So what that means is, at the arterial end, we have a bulk flow or a net flow of fluid being pushed out. And then when we get to the venous end, we have a net flow of fluid being pulled back in. And the reason why this is important is because the fluid that's on this end is handing over oxygen and substances like glucose. And then it's continuing past. And then on this end, the tissue is handing it carbon dioxide and waste. And why is that perfect? Because this is where the fluid gets pulled back in and can get taken back to the heart. Perfect. Perfect, perfect, perfect. Now, the push out and the pull in have names which you need to know. Okay? So, the push out, remember I've drawn it in red. So the push out is called the hydrostatic pressure. Basically, it's blood pressure, right? Because it's the pressure of the blood being pushed to the walls.
So remember that hydrostatic pressure, aka blood pressure.
Of the anterior end of the capillary. Okay, the pull in, which I drew in blue, so I'm just going to draw the blue arrow here. The pull in is called colloid.
Colloid is just stuff. When you think of colloid, it's little particles, glucose, proteins, whatever stuff is there that isn't liquid. Colloid.
Osmotic pressure. And I told you it's the pull.
So they're opposing pressures.
Of the proteins in the blood pulling that fluid back in. Now, let's just think clinically for 1 second. What if we were to increase the size of these gaps in the capillaries? Let's say they're now big enough for proteins to move out. So that means that as the blood.
What happens if the proteins start to leak out? They're no longer trapped in here. What's going to happen?
If the proteins leak out?
Yeah, if the proteins are no longer trapped within the vessel, what's going to happen?
Do they get absorbed?
Probably not. They're no longer trapped in the vessel. What's that going to do to the osmotic pressure within the vessel?
Fluid overload. In the
Where, Zena?
Outside of the cells.
Outside of the cells, and it's not going to get reabsorbed at the other end. Does that make sense?
Yes.
Comes through 30 millimeters of mercury pressure pushing out, not just fluid comes out, but also the proteins come out as well. If the proteins leave the capillaries, is there any pull back in? No. So that means we have all this bulk flow out, including proteins, and there's no proteins to move to the venous end, so there's no pull back in, which means fluid just continues to accumulate outside the capillaries in an area called the interstitium, meaning between the tissues. This is edema. So when you see a patient who has fluid buildup.
So what's edema? What he just said, what is it?
It's fluid buildup. Usually you can notice it in their feet and ankles.
Why is it happening?
Because your heart and everything's not working properly.
Are you sure? What did he just say, Becky? What did he just say?
Well, my internet cut out, so I didn't hear exactly what he said, but
she's like,
I do know from working on a cardiac unit that it means like something's just not working properly. Your body's not getting rid of the fluid and you have a fluid buildup. And a lot of times it has to do with your heart.
Well, yes. And the hydrostatic pressure is too high. That's one way that you'd get edema. Because if you build up the water pressure in here too high, yes, you'd get edema that way, right? Because if the driving force is much higher than 30, it's like 40 or 50, rather than just having a 10 millimeter driving force out, you now are looking at 30 millimeters of mercury instead of 10. Does that make sense?
Yeah.
And so, but if you have proteins leaking out of these vessels, then it's probably not so much it could still be the heart, but it's not so much the heart. It's more the fact that there's just no proteins left to bring the fluid back in. Does that make sense?
Yeah.
I'm sorry, I have a question.
Go.
Is that what ketoacidosis is then?
Mom, can you answer her question? Ketoacidosis, does it have to do with edema? Can it result in edema?
Well, with the proteins leaking out is what I was thinking.
With the proteins leaking out of the capillary beds into the interstitial tissues. She can't really call that up. I would say no right now. Let me think about it a little bit. The reason I called on my mom there was because she was a diabetes nurse practitioner for a long time.
Mm-hmm.
So I want to say no right now.
Okay.
But wait on it. Hold on to it. Hold on to that idea for a couple weeks here, Zena.
Okay. Thank you.
You're welcome.
Pull in is called colloid osmotic pressure. And I told you it's the pull of the proteins in the blood pulling that fluid back in. Now, let's just think clinically for 1 second. What if we were to increase the size of these gaps? Let's say they're now big enough for proteins to move out. So that means that as the blood comes through, 30 millimeters of mercury pressure pushing out, not just fluid comes out, but also the proteins come out as well. If the proteins leave the capillaries, is there any pull back in? No. So that means we have all this bulk flow out, including proteins, and there's no proteins to move to the venous end, so there's no pull back in, which means fluid just continues to accumulate outside the capillaries in an area called the interstitium, meaning between the tissues. This is edema. So when you see a patient who has fluid buildup at the bottom of their legs or at the periphery, this is edema. And it can be due to one of the reasons, a widened gap between the endothelial cells of the capillaries. Think about inflammation. Inflammation is just injury or damage occurring to vascularized tissue. Everyone's had some form of inflammatory response. Let's say you've cut your hand. What happens? You've got the 4 cardinal signs of inflammation. You've got the redness, you've got the swelling, you've got the heat, and you've got the pain. Okay, these have been known for thousands of years. But what's one of those that we need to remember? Swelling. So you cut yourself, you always notice swelling at the site. Why? Because when you damage cells of your skin, these cells explode their guts, and inside there's histamine. Histamine is a chemical that creates larger gaps in your capillaries. Why? Why would we want this? Because if I were to cut myself, whatever cut me, let's say it was a nail, what if there was bacteria on that nail? I now have bacteria in my interstitium. Okay? How am I supposed to attack it and kill it if all my white blood cells are in my capillaries? Well, histamine opens up the gates. And my white blood cells can come out and attack the bacteria, kill it off. But what that also means is fluid comes out. But this is good as well because the fluid sort of washes it away and dilutes it. Okay? Now, you may think that of all the fluid, under normal situations, all the fluid that gets pushed out on the arterial side, that it all gets pulled back in. That's not the case. There is a small percentage of this fluid.
That small percentage of fluid that doesn't get reabsorbed here, what's going to take care of that? What system is going to take care of that fluid?
Lymphatic.
Awesome. Yes.
That once it gets pushed out, it cannot get pulled back in. Now, this is enough, even though it's a fraction of the fluid, it's enough that at the end of the day, if we could not find a way to pull that fluid back in, if we could not pull that fluid back in, our blood pressure would drop enough that we would die at the end of one day. So it means that of the fluid that can't get pulled back in, we do have a mechanism. We need to get that fluid back. How do we get it back? The lymphatic system. So we have lymphatic vessels as well around it.
Okay, you guys, I'm going to end it there. I will post that video. He does a really good job, and I didn't realize. I have never watched it. I just know that he tends to do a good job. I will post that video, and so you'll have access to it. I'm not going to go through all those pressures again. He probably does a better job than I do explaining them, so I'm not going to try. At least not today. I'll summarize it when we go through it in lecture, but he does a much better job. Hydrostatic pressure promotes filtration because it's pushing the fluid out. Osmotic pressure is absorbing it, bringing it back in. You guys, I don't know if you're like me right now. I'm a little brain dead. And let's take a few minutes and walk away just for a second, just for like 5-10 minutes, and come back.
Thank you. All right,
Okay.
We'll see you in a few.
Okay.
All right, I'm gonna have to tag out. I have to get myself going and all
Be
that stuff.
safe,
So.
Becky. Be safe.
Thank you.
Okay?
Hope everybody has a good weekend.
You too, Becky. Be safe.
Thanks,
Thank
you
you.
too.
See ya. Thanks, Casey. Bye.
While he sleeps through the pain. Will that help? Oh, really? Probably is not the right one. It's still the gross. Love you, Jay. Oh, that's fine. Yeah, you tell them, Mrs. You tell them. Did you tell them? Yeah, you tell them. Go get them. Go get them. Did you tell them? Go get them. Go tell them. Did you tell them? Yeah, of course you did. Did you tell them? Yeah, of course you did. Let them know how it is. All right, let's come back to this. So like I said, I'm not going to go through everything that he just said because sometimes they do a better job than I would explaining things. But net filtration pressure, NFP, is the differences between the hydrostatic and the osmotic pressures. Bulk flow, meaning what's the net movement. An edema, when the reabsorption at the venous end is not as strong. And therefore the fluid is not being pulled back into the vessel. Some additional terms. Hemodynamics is what we're talking about when it comes to blood movement. Vasomotion includes vasoconstriction and vasodilation. Bear with me here. Vasomotion, meaning vasoconstriction, vasodilation. By definition, that's what it means. Vasomotion is the product or the sum of vasoconstriction and vasodilation of the blood vessels. Vasotone, the amount of vasoconstriction. Elastic recoil. So this was what I was getting at with the elastic arteries. This is the amount of recoil you vasal recoil. Vasorum, kind of an interesting word. Vasorum, meaning the blood vessels that are in the adventitia, the tunica externa or tunica adventitia, that supply the blood vessels. So the blood vessels, the mini micro blood vessels that supply the blood vessels. Questions? So endothelium, tunica intima, endothelium, subendothelial layer. One thing that we have in arteries that we don't have in veins. So one thing that we have in veins that we don't have in arteries are valves. Just like the valves in the heart, these are going to prevent things from moving backwards. Lauren asked about spider veins. What spider veins are, or varicose veins, are when these valves give out. Why does it exactly occur? I think it's a combination of genetics and other factors. I can say that I started getting them at about 15. And I got them, I think the major reason I got them was because I started working out, not excessively, but that's when I started cross-country cycling, was about 15 years old. And I think to maintain the muscle mass that I was developing during that time, I developed a lot of spider veins and varicose veins from it. And so I think it's a lot of different things. One thing that they do tell you is it can have to do with dehydration, maintaining your hydration, because that's also maintaining your blood pressure, your blood volume. But I think it's a combination of a lot of different factors. But it's essentially these valves are giving out. They work to essentially fight against gravity because the blood pressure is lower in the veins. And therefore you get varicose veins and spider veins for that reason. Questions? Well, here's some just histology. One major way that you can always tell the difference between, if you're looking at a cross section through a vein and an artery, is arteries maintain their shape as a circular vessel. Veins do not. They tend to have a larger lumen, and don't tend to be a very circular structure in comparison to arteries that are very circular in structure. Arteries tend to have more muscle. Veins have less muscle. And veins also, with their having less muscle, have more tunica externa or tunica adventitia, thicker tunica adventitia. Arterioles and venules right next to one another. Arteries, remember, arteries tend to maintain their structure a lot better. You'll see this one's more oval-like in shape. Arteries tend to be much more circular. Arterials tend to be much more circular because they have the muscle around them to support them. Veins do not. This is a picture right from your manual. There's one part of your questions that asks you to label these. This is that same exact picture with it labeled. But this is a vein, this is the artery. One thing you notice is that the volume inside the veins becomes much greater than in the arteries, because where there's less pressure, meaning the veins, the further you get away from the heart, going through all the pathways of vessels, the lower the pressure becomes. So as a result, how are you going to keep the blood moving forward? You have to reduce the resistance by increasing the size, just like you would a hose in your backyard. So review of arteries, arterioles, capillaries, venules, and veins. So arterioles, vasoconstriction. What does it mean to, if you think about it, what would, if you have a diameter of an arteriole, if you vasoconstrict it, what happens to the diameter?
It closes.
It shrinks. Yeah, it shrinks. So vasodilation, meaning it opens it up. What is going to happen to the resistance in the vessel? Think of it as the same as your water. What's going to happen to the resistance in the vessel if you vasoconstrict it?
More pressure.
It's going to go up. Yeah, it's going to go up. Another thing is, what if rather than having just water, you have syrup? Which one flows faster, water or syrup down a hose?
Water.
Why?
Because of that viscosity.
The viscosity, right. So the radius, vasoconstriction, vasodilation, is going to affect the pressure and the flow. The viscosity is also going to affect it. What about the volume? Well, if you only have a trickle of water running through the hose versus a lot of water running through the hose, it's going to affect the resistance too, right? Does that make sense? So yes. Blood pressure, resistance within the vessel, and blood flow, how do they relate to one another? Flow equals to the change in pressure over resistance. If the pressure at one end of the hose is much greater than at the other end, fluid is going to move a lot faster than when the pressure differences between the two sides of the hose are less, right? If you turn on the hose to water your yard and you only turn it on just a little bit, because the pressure is so much lower at the nozzle, or at the spigot, the water's only going to trickle out a little bit because the pressure, the magnitude of the pressure difference is so low and so small. Over resistance. So if the vessel size is the same and the viscosity is the same, volume is the same, the flow is only equal to the difference in the pressures at one end versus the other end. Does that make sense?
Yes.
Okay. So here's our aorta, our arteries, our arterioles, our capillaries, our venules, our veins, and then our vena cava. The total area, cross-sectional area, much higher for the capillaries because the resistance is, we want the resistance to be higher. Therefore, the velocity is less, is the least amount. Because remember, we want it to, it's only like the red blood cells are only going down one cell at a time down the capillaries because the resistance is so high and the vessel is so small. And therefore the flow is very, very, very slow. Let's see here. The resistance, the velocity is going to be very slow in the capillaries and have a much bigger surface area in the capillaries. Much more capillaries than there are other vessels. It's also why the velocity can be so low. Increased velocity in the aorta because of the blood pressure differences. Just coming off the heart, you remember being able to feel your pulse because of the aortic arch right underneath your finger when you stick your finger behind your mediastinum. The pressure is so high. That's why the velocity is so high. And it drops off as you get into the capillaries where the total area is high because you're going to increase the resistance. Radii, vessel radii, going to increase the resistance. And it starts going down with veins. Vena cava. Vena cava has a very high velocity because the cross-sectional area is exceptionally large. Elastic arteries, muscular elastic arteries. Vessel diameter shrinks down to nil at the capillaries. So here we're looking at vessel diameter. Oh, wait, bear with me here. Different vessels, vessel diameter. Very small for capillaries. this diagram that you were looking up here, cross-sectional area through the capillaries, is the same one. Do you see that? With the velocity of flow. So these are superimposed on one another right here. So vessel diameter, very low for the capillaries, and it goes up drastically with the vena cava because the greater the diameter, the less resistance there is. And we need that because there's very little pressure in the vena cava. And if we don't have a greater diameter, the blood isn't going to flow at all. Questions? So blood pressure very high for the aorta, very low at the vena cava, and intermediate through the capillaries. So pressure is proportional to the inversely proportional to the distance from the heart. And here's the aorta, very high, right next to the heart, and then it drops off. And flow is proportional to the pressure. Remember, systolic blood pressure is the blood pressure right next to the contractions of the heart, right next to the aorta. Diastolic is the blood pressure when things are relaxed. And as you get further away from the heart, the differences between the pressure differences between when the heart is contracting and when it's relaxing are going to become smaller and smaller based on the fact that you're further and further away from the heart with regards to the road lengths or the vessel lengths from the heart. So how do you measure blood pressure? Using a sphygmomanometer. And I'm sure I'm still saying it wrong. Sphygmomanometer.
I can't say it either. It's really hard.
It's a great word. It's a very long word. So sphygmomanometer is how you measure blood pressure. You cut off the circulation just temporarily. And then when you start to actually hear the blood coming through, when you start relieving the pressure, start hearing the blood coming through, that's your systolic pressure. You're hearing turbulent blood flow through the artery that's just opening up. The Korsakoff sounds are those sounds, that turbulent blood flow. As it's squeezing through the vessel that you're just opening up. Once you're no longer hearing it, the last sounds that you hear is the diastolic pressure. So ventricular contraction, ventricular relaxation. Ventricular systole ventricular diastole. This is what the guy was getting at with that video. Mean arterial pressure. Mean arterial pressure is the diastole pressure plus the pulse pressure divided by 3. The pulse pressure is the systolic pressure minus the diastolic pressure. Your venous pressure, or the pressure between your legs to your neck, is going to change. For every 12.88 millimeters elevation. We won't worry so much about venous pressure. We should have just taken it out because it isn't really necessary here. So flow, remember flow is the difference, is proportional to the pressure difference divided by the resistance. So F equals the change in pressure over the resistance. And the resistance, the 3 factors that affect the resistance are is the vessel radius, the viscosity of the blood, and the blood volume. Last thing, what's the relationship between mean arterial pressure and cardiac output? If the mean arterial pressure, see here, I wish I hadn't have asked this question because I'm trying to remember what, let's see her. Mean arterial pressure and cardiac output. I shouldn't have asked this question because I'm not sure I know how to answer it right off the top of my head. Give me 1 second here. If the mean arterial pressure goes down, the cardiac output goes down. Why would that be the case? I remember now. If the mean arterial pressure goes down, why would the cardiac output go down? What is the mean arterial pressure? What does pressure measure? Mean arterial pressure, what is it actually measuring, guys?
Is it the pressure in the arteries?
It is, it's pressure in the arteries. And what is that a measure of? What is that? What is that? Well, since we can't directly measure the flow of blood, we have to use the mean arterial pressure, right? Does that make sense? You can't exactly cut off a vessel, drain it, and see how fast it's moving, right?
Yeah.
So we use the mean arterial pressure as a measure of the flow. Does that make sense? Maybe, maybe not.
Yeah.
It does.
Okay, so mean arterial pressure. Now, what is cardiac output? What's cardiac output?
The fluid being pumped out by the heart.
Per minute, right?
Right.
And you remember, so it's cardiac output equals stroke volume times heart rate. Let me give you an example. So I'm going to go with, I know somebody who frequently can have, their blood pressure can be like, oh, let's say 105. Remember where he said it should be about 120 over 180. 105 over 65. Is that high or low blood pressure? What would you say? Say it again.
Slightly low.
It's slightly low. And this same person has a heart rate of, oh, let's say 100. What does this say? When you have a blood pressure that's 105 over 65, and you have a heart rate that's 100, what's that? Is that heart rate, is it high, is it low? What would you say?
Heart rate's a little high if it's resting. Well,
yeah.
Is it? Yeah.
Yeah. So what that means is that their stroke volume is really low. Do you see that? You remember that from last week? Their stroke volume is really low. So if you can bring this up, their stroke volume will go up and their heart rate can go down. Do you see it? That is the relationship between mean arterial pressure and cardiac output. Questions? I think that's the end of this lab. I do want to spend a minute going through some heart stuff. Let's see if we can do just a little bit more. No, let's not. Never mind. Let's wait until next week, because once we start into it, it's going to take more than 10 minutes. Questions for me? Actually, no, let's do a little bit. I'm sorry, bear with me. I'm thinking back on the lecture PowerPoint. Let's do just a couple minutes of it and then we'll give up with it for today. You can't take my teaching assistant. You can't take her. You can't take her. My mom's getting ready for bed, so she's going to try to take my teaching assistant. She says, I don't want to be with you anymore.
And they're all saying they want to go to bed in the middle of the afternoon.
Does your mom work nights or does she always?
No, she's retired. She's retired, so she can sleep whenever she wants.
Oh, okay. Good for her.
Oh, and she's been sick for like 2 weeks, 3 weeks now. Yeah.
Because everybody COVID just wants to sleep all the time.
I know. Well, and she finally went and saw a doctor and she's had 4 COVID tests. So it's not COVID, but it's something else that's just not. The doctor thought it might be a sinus infection of some sort, but it's been really quite nasty. All right, let's go through just a little bit here and then we'll quit for today. So you remember when we talked about action potentials, or when you talked about action potentials in 121, how did they work? How did action potentials work? How did action potentials work?
Don't they sense something and then they send signals to the central nervous system, and then the central nervous system like sends out message to the receptors and then they act upon that?
Yep, kind of like that, but it's more an ion flow. So sodium is going to move in, potassium is going to move out. And it's an ion flow thing. And one thing that we're going to learn about the heart is the heart has its ability to initiate its own action potentials. The autorhythmic cells, we have 2 different types of heart muscle cells. We have autorhythmic cells that are going to initiate and conduct action potentials that are going to be responsible for the contraction of the working cells, the cardiomyocytes. So autorhythmicity, the heart will contract rhythmically as a result of action potentials it generates by itself. The autorhythmic cells are going to initiate an action potential, which is going to be responsible for initiating an action potential in the contractile cells. And it's a matter of depolarization, just like we saw with action potentials. It's a depolarization and a repolarization. You remember that from before? Sodium high depolarization, potassium repolarization.
Yes.
So 2 different types of action potentials in the case of the heart. The autorhythmic action potentials, and then we have action potentials of the cardiac cells. One thing that the reason that the heart is autorhythmic is because it does not have a resting membrane potential. If you remember, there's what the resting membrane potential for the nervous system is like minus 70. And it stays down that way. It's almost straight until you start to get inhibitory and excitatory postsynaptic potentials. Here, it's not the case, it's rhythmic. And there's no resting membrane potential, no constant resting membrane potential. So there's a slow depolarization that just every time that repolarization occurs, you get another slow depolarization that starts all over again. And it keeps occurring every time. So rather than have it just be straight line and flat line until you start going up to and reaching threshold, it's moving in a rhythm. So there's a slow depolarization due to the opening of what are called funny sodium channels. They're different types of slower sodium channels. They're slow to open. So they open, and then some calcium channels start to open. And then at threshold, which is minus 40, the calcium levels skyrocket. So rather than dealing with sodium channels, we're dealing with calcium channels. The calcium voltage channels open up drastically and calcium starts pouring into the cell. Up until we get to just about 0 millivolts. And then those channels close and the potassium channels open. Do you remember this from before? So same thing here, but rather than having sodium right here, we have calcium. And then the potassium channels close. The funny sodium channels start to open again. And we start all over again. Do you see that? See how it works? And now I will say that I will see you in a week.
All right, thank you. Bye-bye.
Oh, next Friday.
Thank you.
You're welcome.
Thank you.
Oh,
Thank
Miss
you.
Mary.
Yes, we need to talk, Casey.
Yeah, do you have a minute or no?
Yeah, give me one second.
Okay, take time.
Oh, little boy, you're all right. Casey, I'm going to stop recording just a sec.
Okay.